So hi everyone, welcome to our webinar. So today we're doing a special webinar, so it's Meet the Expert. So we're going to be speaking with Dr James Quinn, who's going to be presenting the advantages of fluorescent Western blotting for dementia research and beyond. And just want to let you know that if you have any questions for James, then feel free to leave them either in the Q&A section or in the chat section. There's also an opportunity, if you want to unmute yourself, you can just raise your hand and I can unmute you as the controller, and then you'd be able to ask James a question as well. So if you want to move on, James. So I'm just going to introduce James. So James is currently a neurology research fellow at Massachusetts General Hospital, which is in Harvard Medical School in the USA. He has, at the moment, 8 publications in dementia with many more on the way. James did his PhD at the University of Manchester in the UK, and that was after his BSc at the University of Sussex. James is a huge advocate of public engagement and outreach. He's also incredibly active on Twitter, sharing all the latest news from his research and also that within his field. So I do encourage you to follow him if you also want to engage, and contact him after the event as well. And outside of science, James is also a massive fan of anything outdoors, loves to go hiking, exploring, and all sorts, and would love to travel the world, especially hopefully once travel corridors open and we're able to go traveling again. Anyway, so that's it for me, and I'll give you a quick chat about some, if you stay behind after James's talk, I'll be able to tell you about some of the giveaways that we're doing at Protein Tech, but that's it for me, so on to you, James.
Thanks for the introduction there, Rebecca. So I'll just start talking a little bit about what I'll be talking about today. So I think the first thing we're really going to cover is the difference between fluorescent Western blots and enhanced chemiluminescence, otherwise known as ECL, which is what I did all my PhD on, but have now kind of converted to fluorescent Western blots. Kind of important tips for performing fluorescent Western blots, focusing on 3 main applications. So the first application is looking at tau biology and Alzheimer's disease. This is from the research from my PhD, and then applications 2 and 3 are from my postdoc. So one is looking at neuropeptide biology in Alzheimer's disease, and the third one is looking at TDP-43 biology in amyotrophic lateral sclerosis, otherwise known as ALS. And we can sum up with some conclusions and questions. So feel free to put them in the Q&A, as Rebecca was saying earlier. And then she'll summarize with a kind of a note from Protein Tech at the end. And obviously, thank you for Protein Tech for kind of sponsoring this presentation as well. So I'll give a little bit of an overview about Western blots. So obviously, the first thing you do is you want to get your proteins. So isolate the proteins. So basically prepare the lysate or media, or if you're doing a different fractionation experiment, or kind of however you're trying to get your protein to start the experiment. So you'll do your prep first. And then the first thing you'll do is you'll do protein separation by SDS page. And essentially you go from your negative to your positive charge, and based on the percentage of the acrylamide in the gel, that will dictate how fast the protein separates. So basically, if you have kind of a low percentage gel, you're kind of looking at high molecular weight species, and if you have a higher percentage gel, you're looking at lower molecular weight species. Then you take your protein and transfer it onto the membrane. This can be done with like a transfer cassette. Then you block your membrane in a container, incubate your primary antibody, and then you wash off the primary antibody. People tend to do primary antibody overnight, and then you can do your secondary antibody and you wash that off, and then you image it via chemiluminescence or by fluorescence. So on the left we have a chemiluminescent image, and then on the right we have a fluorescent Western blot image. So this is a kind of summary over the difference between fluorescent Western blots and ECL. So ECL is a light-based reaction. So you've got your target protein, which your primary antibody is bound. Then your secondary antibody has a HRP, horseradish peroxidase conjugate on it, which converts the substrate is luminol, and then it converts into oxidized luminol, and that emits light. And that's what is detected on the blot that you can see back here. However, fluorescent Western blots, these work differently, where you've got your antigen, and then kind of that is what's being reacted to, and then you've got your rabbit anti-antigen here, or you have a mouse anti-antigen here, or goat anti-antigen here. So this is for rabbit, this is for mouse, this is for goat. So obviously you can see this is allowing you to multiplex kind of different antibodies to different antigens in the same experiment. And then finally, your secondary antibody is kind of directly conjugated to a fluorophore. And you can see that with the different wavelengths of the light, you can detect them all within the same image. So I think you can tend to do between 2 and 4 different antibodies as long as they're raised in different species. So obviously rabbit, mouse, goat, rat, really any species you can name, there's relevant secondary antibodies to those species. So obviously some advantages of fluorescent Western blots, it allows you to multiplex different antibodies. So for example, your protein of interest, as well as a total protein stain within the same blot, which is great for quantification. The stability of the fluorescent signal over time, you can basically take the membrane, leave it in PBS, leave it in the fridge. And if you come back 2 weeks later, you will still get really good fluorescent signal, whereas that would never happen with a kind of ECL-based reaction. Once you've added your ECL, that signal decreases very quickly over time. After secondary antibody, you can just wash your membrane, place it directly into the imager. There's no need to add, for example, with ECL, you don't have to mix the 2 reagents together and then add it to your membrane. You don't have to use film, whereas a few people in my lab, they have to like run upstairs to the floor above to take it to the film imager, whereas I'm just like, oh, okay, got my membrane, put it directly into the imager. You basically have an improved dynamic range versus chemiluminescence, more reliable and reproducible data when it comes to quantification. And you can always convert back to black and white images if you don't want to have the kind of the color image. So the first application of using fluorescent Western blots that I've been doing in kind of my PhD was looking at tau biology in Alzheimer's disease. My whole PhD was on Alzheimer's disease and really focusing on Alzheimer's disease. Was focusing on the protein tau. So here we've got a healthy brain. This is a microtubule in blue, and then you can see that tau is kind of on the structure of the microtubule, and it acts to stabilize the microtubule. Obviously, microtubules are super important. They act as a highway within the cell. It allows transport of organelles, and it basically acts to kind of hold everything together. And you can see the phosphorylation, which is recognized by this P, basically kind of a normal level of phosphorylation keeps the tau bound to the microtubule, keeps it stabilized. So you've got a healthy neuron over here. And then down below, in Alzheimer's disease brain, you can see you've got the degeneration of the brain. They lose approximately 10% of the brain mass in Alzheimer's disease. And you can see here that the microtubule is disintegrating because you've got a hyperphosphorylation of tau. You start to form these tau protein tangles, which kind of tangle together to form these neurofibrillary tangles, which you get lots of aggregated forms of and phosphorylated forms of tau. This leads to the death of neurons. You also get fragmented species of tau as well, and I'll talk a little bit about those later on. So this experiment I wanted to look at was whether tau phosphorylation is increased in different fragments of tau compared to the full-length version of tau. This work's in preparation. We hope to kind of get it submitted this year. And in this experiment, we took HEC293 T cells. We transfected them with an empty vector, the full-length version of tau 1 to 441, and then 2 C-terminal fragments that we were interested in, 195 to 441, 210 to 441. People who know a little bit about tau know that the aggregation-prone species of the aggregation-prone region of tau sits in the C-terminal region. So somewhere within the range of around the 300 to 330 mark amino acids. And essentially, you can see that in this experiment, I wanted to compare the full-length tau against the 195 to 441 and 210 to 441. In this experiment, we also want to then compare it to the level of total tau that is being expressed in the cell. We noticed that we were getting different levels of expression between when we overexpress the C-terminal fragments as well as when we overexpress the full-length version. So this was a great use of fluorescent Western blots because you wouldn't be able to do this experiment with ECL because it would require you to do stripping and reprobing of the membrane, which has caveats, or then run 2 Western blots. One where you would do your K9GA, which is a total protein stain for tau, and then PHF1, which recognizes 2 phosphorylated tau epitopes, serine 396 and serine 404. And in this experiment, we're able to quantify that the two C-terminal fragments of tau have an increased level of phosphorylation compared to the full-length version of tau. This was super interesting for kind of our data, and then we can also make this equivalent to actin. So in this experiment, it would have been also nice to have done that third that third multiplex. So you could have done the K9JA, the PHF1, and then done actin, and you could have had all of them on the fluorescent Western blot. I don't think the imager we had during my PhD had that capability, but a lot of the images that are available now do have that capability. And here you can see, obviously, we're able to quantify directly. So basically draw kind of a square around this band, square around this band, square around this band, and then make it all relative to the amount of the total tau that is being expressed. So the next application is kind of the work that I've been doing in my research fellowship here at MGH. So again, we're looking at Alzheimer's disease. So here on the left with the healthy brain, on the right, an Alzheimer's disease brain. And we're really interested in neuropeptides. Neuropeptides are a signaling molecule that travel between synapses. They have super important roles in essentially just maintaining normal synaptic activity. They're distinct from neurotransmitters. So neurotransmitters have, for example, reuptake mechanisms, whereas neuropeptides do not have these reuptake mechanisms. They are essentially released and they go from synapse to synapse or synapse to microglia. The only known mechanism for getting rid of the neuropeptides is with proteases that cleave the neuropeptides in the extracellular space. We're really interested in this granin family of neuropeptides, chromogranin and secretogranin, which have really important roles in neuronal and glial function. We have chromogranin A, secretogranin 1, secretogranin 2, secretogranin 3, secretogranin 5, and VGF. VGF is also known as secretogranin 7. People also know secretogranin 1 as chromogranin B. So you know these families are extremely similar. And we're really interested in these because we've shown in some work that we're hoping to get published soon, where some of these neuropeptides are decreased in the cerebrospinal fluid and brain tissue of people living with Alzheimer's disease compared to healthy controls. So it's kind of a potential marker of synaptic health in Alzheimer's disease. So they're really exciting potential markers that we're developing at the moment. In order to kind of get at why these neuropeptides are decreased in the CSF and brain tissue, we are kind of going back to basics. So we are looking at kind of the cell biology of these neuropeptides and seeing what's going on. So this is a great experiment in order to look at essentially just overexpression in these cell lines. So here we've overexpressed VGF in HEC293 T cells. So this is in lysate and this is in the media. So here in the lysate, we have VGF overexpressed in these two, and then we've done the protein tech VGF rabbit antibody, which is in the green, and then the GAPDH in mouse is in red. Now you can have a mouse, I think the rabbit is anti-800 and then the mouse is anti-680, but you could obviously flip that, it's just based on what secondary antibodies you buy. And then here we've got GAPDH antibody in red. So you've got your total protein stain here in red, and then you can flip it to the black and white. I always like to flip it into the black and white because sometimes you can see extra bands kind of popping up, but the kind of the dual color is also really helpful. You can see we've got really nice overexpression of VGF, both in the lysate, and then we're seeing a lot of this VGF being secreted. So it makes sense based on we're showing that these proteins are secreted from the cell and they're spreading kind of across the synapse. The third application is studying TDP-43 biology in ALS. So we've got this collaboration with AC Immune, a company based in Switzerland, to really develop TDP-43 as a potential biomarker of ALS. And essentially, I'm kind of trying to go in and really study these antibodies against TDP-43. So we really want to develop a model in order to overexpress TDP-43 to kind of analyze how well these antibodies are able to recognize TDP-43. In ALS, you have TDP-43 mislocalization from the nucleus to the cytoplasm. So in blue, we've got TDP-43. So here in the nucleus and then into the cytoplasm of the neuron. And then it starts to aggregate. So you see we've got more and more species kind of spread throughout. And then this leads to the motor neuron degeneration that takes place in ALS. So in order to kind of study why this is taking place and to study the antibodies as a potential biomarker, we're really just trying to understand what species of TDP-43 these antibodies are recognizing before we then translate that work into cerebrospinal fluid, blood, brain tissue and muscle fiber extracts from patients who had died from ALS. So here, again, this is a very similar experiment where we're looking at TDP-43 with the protein tech antibody and then GAPDH with the Thermo Fisher antibody. And you can see we've got really nice TDP-43. So this is a TDP-43 M-cherry vector. So the TDP-43 runs about 72 kilodaltons. And then this is the endogenous form of the TDP-43. So the SHSY5Y cells are a neuroblastoma cell line, and they have really good endogenous expression of the TDP-43. So we can see both the overexpressed version up here, as well as the endogenous version down here. And this is the kind of TDP-43 channel in black and white. And again, we're using the GAPDH as just a marker of the total protein. And then again, here, this is the media. So in the media, you can see we've got some level of secretion of the endogenous form, but we're not really seeing this overexpressed version being secreted out the cell. So we're really just using this as a way of kind of studying these AC immune antibodies, but I'm not going to be showing the data for that. And I thought I'd just wrap up with some bad Western blots. So the Western blots I've shown are really nice. And these are a few Western blots that have gone horrifically wrong. And I'm just going to talk a little bit about why they've gone wrong. So the one on the top left was one actually I got really recently. Here we've done an IP of TDP-43. I'm not going to go into explain the different lanes and stuff, but essentially it was the last gel we had in the box, and I needed to get the data kind of ASAP. And I opened the gel out the box and I saw it had a few air bubbles in the gel. We normally get ours kind of bought commercially. I used to make them during my PhD, but now we just buy them in. I noticed there's a few air bubbles kind of in the center region. And you can see very clearly that these air bubbles have affected the way in which the gel has run. So you've got this kind of nice aberrant pattern of gel, but you can see that the IP has worked. You can see the TDP-43, but it's not a perfect blot. So I'm actually repeating it today. The experiment below was done by my undergrad student in the lab right now, where we ran some plasma, again, to look at TDP-43, but we did not dilute the plasma. So anyone who's done any experiments with plasma knows that plasma has extremely high protein concentration. So when you don't dilute it, this is the recombinant TDP on the left, and I think this is some lysate input from SH cells, and then this is just an absolute mess. And I remember when she was loading it, it took her about 30 minutes to load the sample because it was just so gloopy. And you can see very clearly this is coming through. This one here in the middle, there was an ever so slight, I think there was a tear in the gel at the top here. So when you then transfer it, you can see the very clear break here in this band, and then a little break here in the ladder. This one down here was essentially, we just put the membrane into too small of a box. So it actually folded ever so slightly on the left. And this is just a base, you don't want that. You want to have your membrane completely flat. And then the one on the right, we were getting some ghost bands, and we kind of realized that this is a mixture of a few things. This is a mixture of our transfer cassette not being tight enough. So we're not getting kind of efficient from the gel onto the membrane. And also this was due to not having high enough expression of our protein of interest that we were trying to study, right? So we were just putting too little protein onto the gel. So the fluorescent imager really struggled to kind of pick up the correct bands for that. So yeah, the gel is kind of, the bands are nowhere near where you'd expect them. These are the right bands, but they should be here, but they're somewhere all the way over there. So yeah, my whole PhD was on doing Western blots. My whole postdoc's doing Western blots, but they still go wrong. But it's good. You can kind of start to troubleshoot kind of why they've gone wrong. So some top tips for performing Western blots. Obviously, don't do any of these. But here we have obviously the first tip. We kind of realized that no one else was doing it with our fluorescent Western blot scanner, but it's super important to clean it. So it actually states to clean the fluorescent Western blot scanner with isopropanol, but just check what chemical is required to clean your Western blot scanner. And use methanol and water to clean all of the equipment used for the transfer cassette. We noticed that I remember I spoke to Lycors, the customer service from Lycor. I'd always recommend reaching out to the customer reps at different companies. They all have experts within these companies to help you out. But I spoke with the company rep from Lycor, and he looked at some of the blots I was getting, and he was like, it's clear the equipment you're using is dirty. And he was like, clean the sponges with methanol. And I remember I did it, and the methanol went from being kind of clear to like, it was like a bright orange yellow color. So ever since then, I've been really on it with like cleaning our equipment probably every 1 week or every 2 weeks. Keep membranes protected from light after adding fluorophore. We bought these pink boxes, which are great. These are like pink Lycor boxes. Everyone else has these black boxes, but you can clearly see which ones are ours compared to every other lab, because we've got these pink ones that really stand out. But essentially, you just want to make sure there's no light kind of getting in after you add the fluorophore. We found that nitrocellulose works a lot better than PVDF for fluorescent Western blots. You can buy, I think PVDF has a higher fluorescence, like a background fluorescence. You can buy like low background PVDF membranes, but we found just buying a big roll of nitrocellulose, cutting it up works perfectly for our kind of our function. We found that using the vendor supplied antibody diluent and blocking buffers works really well. And really important to take advantage of that ability to multiplex. You can test out 2 to 4 different antibodies for the same protein. So sometimes you could even do like a mouse antibody to your target protein and then a rat antibody to your target protein. If you want to see which one works better, you can do that in the same experiment. And important to take some time to optimize your signal-to-noise ratio. I'd always recommend testing out a few different, for example, the TDP experiments we were doing, we always include a recombinant TDP control, but we did experiments. We tested 1 nanogram of recombinant TDP 43, 10 nanograms of recombinant TDP 43, and then just found out where kind of the optimal ratio of that is. There's a few really good software online that you can use to kind of optimize these things. So in conclusion, fluorescent Western blots are extremely versatile and allow for antibody multiplexing. Tau phosphorylation is increased at specific epitopes in C-terminal tau fragments compared to full-length tau. We are able to overexpress neuropeptides in HEC neuropathic T cells. We can also do this in S8-as-Y fibroblast cells, and we're potentially moving into iPSCs further down the line. For further characterization, TDP43 can be overexpressed in S8-as-Y fibroblast cells, even though they have a lot of endogenous TDP43, to look at its dysregulation in ALS and to develop these kind of novel diagnostic biomarkers as well. And all of this can be studied and more with fluorescent Western blots. So I hope this gives you a bit of food for thought to start thinking about different ways you could use fluorescent Western blots in your research. I'm a big fan. Doing 3 years on my PhD, just having my whole thesis filled with ECL-based reactions to then move into the fluorescent Western blots has been a real lifesaver for kind of the work I'm doing right now. So I'd just like to thank a few people. So obviously special thanks to all of our donors and the families. I didn't include any, well, we did include some of the blood that we were getting from patients with ALS, but obviously super important. We're using blood, we're using cerebrospinal fluid, we're using brain tissue from patients. The work was completed with funding from Bright Focus, Target ALS, Yale, NIA, and Challenger Foundation, and I used BioRender to generate a lot of the figures here. And I want to thank a few people. Dr. Stephen Arnold and Dr. Becky Carlisle are both my bosses at MGH. Bianca has been a great help in the lab, and Liz is my student who's doing fantastically right now. And then Savannah was a tech in the lab who's now doing her PhD at Yale. So yeah, that's it. I see we've got a few questions and I can let Rebecca ask me them.
Great, thank you for that excellent talk, James. So we've got one question from Julie and from Irina, sorry. Can fluorescent Western blotting help produce background noise on the membrane? She says there are multiple bands around the band of interest, and she just imagines the answer is no, but just wondering.
I think that's something that ECL can be really good at, and you can definitely do the same thing with fluorescent Western blots. You can really play around with the length of exposure, and you can also play around with the brightness and contrast really well. So it saves all of the raw data, like every time you take an image, right? And then you can go in on that image and you can alter the brightness and contrast. So I'm always interested, a lot of our work is looking at fragments of proteins, right? So we always want to kind of really pull up the exposure after we've collected the image, altering the brightness and the contrast, just to make sure we can see those lower molecular weight species, which sometimes aren't as abundant as the full-length version of the protein. So I think I would actually say it's kind of more user-friendly than ECL, because you really have all of your raw data there, and you know that if you play around with the brightness and the contrast, you can always go back to the original as well.
Thank you. And Julian says, how do you perform the protein extraction from nervous tissues? Obviously, that's quite a long question, James, and if you do want to just email these people separately, that's fine. But if you have a brief overview.
Yeah, so with CSF, I can start with CSF. I mean, it's kind of nerves. Yeah, it's released from nervous tissue. CSF is really easy. We don't need to do anything. We just add loading buffer to it, boil it, then we can load it onto a gel. Something like brain is obviously a lot harder. Brain, we tend to do homogenization. So you can basically use a kind of a homogenizer. I'm trying to think of the better word for it, but it's essentially, it goes into your sample and it sonicates your sample by basically just vibrating the sample, and you can sonicate 10 to 15 seconds, and that can get your brain tissue into solution. So obviously you just have to make sure you're adding the right buffer. Something like tau and TDP-43, a lot of people are interested in like the aggregated forms of the protein as well. So there you have to do different fractionation. So during my PhD, I did a lot of like sarcosyl and soluble fractionation steps. So there we use sarcosyl, which is a really strong detergent, to pull out the super aggregated forms of tau, for example.
Thank you. And Nina says that she is a total fluorescence convert, but she just wanted to ask about your choice of membrane and if you'd ever looked at nitrocellulose versus that PVDF fluorescence membrane yourself.
Yeah,
and
so I
what
did
kind
do
of kit to use the transfer, sorry.
No, that's fine. I did try. So there was one time we ran out of nitrocellulose membrane, and I was like, oh no, what do we have lying around in the lab? I think that was when there was a lot of issues with getting orders in, especially around COVID, when we kind of came back into the lab. I was like, okay, what else can we use? So I tried PVDF and it just did not work anywhere near as well. We saw a lot more kind of background fluorescence. I did try in my PhD the low background PVDF membrane, and that worked pretty much the same as nitrocellulose. But it costs more money, right? So I remember we were like, okay, nitrocellulose is clearly cheaper. Kit for transfer, we just use the generic transfer cassette. If I could, I would buy one of those ultra fast transfer machines that can do it in about 10 minutes, but they're quite an expense. So I'd definitely look at getting one of those in the future. But yeah, we just use, I think we use Biorad for our transfer system.
Akshay asks, how can we improve the signal for proteins which have low expression?
That's a great question. I would just load as much of it as possible into the gel as a first thing. Anything you can do where you have I think the best tip is always have a positive control on your Western blot, right? Like you see with TDP-43, we're always loading recombinant TDP-43 onto that gel, and we know that the antibody can react with that. And I think with any imaging, it's so much easier to find your band of interest when you've got a recombinant standard on there. Even if you have to like massively put up the exposure, it's basically just one black line, right? But you can then just about see your band down there. That will make it a lot easier. You can also try things like increasing the length of increasing the length of imaging. So imaging on the highest setting. So I know our LICOR has like a low, lowest, low, medium, high, highest. Don't fully know exactly what each of those settings does, but we were like, if it's important, we do high. If it's not that important, we do low. And just kind of play around with those different settings that you can use. But anything where you can put as much protein on the gel, always have a recombinant standard on there, and then play around the different settings on the LICOR or whatever machine you've got. It's definitely helpful.
Great. And Julian asks, is it necessary to use an additional buffer for fluorescent Western blot compared with the traditional ECL?
Could you go with that question again, Rebecca?
Yeah, I'm not entirely sure either. He says, is it necessary to use an additional buffer? So maybe he means a different buffer for fluorescent Western blot versus that ECL.
Oh, no, I see what you mean. So we keep everything the same as you would for ECL, but the only difference is that we use like the LICOR blocking buffer and the LICOR antibody diluent for the block stage, and then everything else we just use PBS, 1 times PBS, 1 times PBS tween. I do 4 washes every 5 minutes, so wash one at 0 minutes, wash one at 5 minutes, etc. with PBS tween, and then 3 washes in PBS tween after, and then a wash in PBS. But yeah, the only thing we buy is the antibody deodorant and the blocking buffer. I would recommend using the vendor supplied one, but you could probably test. I haven't tested out not using that and just using BSA or milk for the blocking buffer. But I think once you've got something working, it's best to kind of keep that consistent, especially when it comes to publication. It's important to be able to state like what you dilute your antibody in and what blocking buffer you used.
I've got a few more questions, but these are really great questions actually. So David asks, how much protein did you load in the well for the fluorescent Western blot? I suppose it varies in the experiment. And does it need less total protein than conventional ECL?
I would say it's pretty even when it comes to the conventional ECL, but I mean, we've loaded recombinant proteins at like 10 nanograms and it can get detected. So we've gone like really low for some experiments. So it has really good detection. And I think the biggest thing for me was like, I just didn't want to go upstairs and use the film imager. I used film in my undergrad and I was like, I don't want to use film again. So we had like a nice ECL scanner in my PhD, but here at MGH, there is a fluorescent Western blot scanner like on the room. So it was on my floor in the lab. So I was like, I'm going to learn to use this, right? So I haven't done too much optimization of this versus ECL, but I know we have done some like dot blots as well, where we just like dot on really low amounts and we can get detection from those.
Thanks. And another really great question from Lionel, which is saying, do you ever encounter issues of cross-reactivity when you miss 2 or 3 primary antibodies on your membrane? And he says, essentially, would an antibody bind non-specifically to another protein and shows the fluorescent signal that is not specific?
Yeah, definitely. We definitely get a lot of non-specific binding. So it's always really important to characterize your antibody. That is like I can tell you some of we've actually found the protein tech wants to be pretty good, but some antibodies are just, we had this massive antibody spreadsheet in the lab because we're interested in 6 neuropeptides. I would say for some of the more conventional neuropeptides like VEGF is really well studied. That was super easy to find antibodies for. Now some like secretogranin 5, we must have tested, and secretogranin 3, we must have tested like 10 antibodies for each of those and found that they either don't react with recombinant forms of the protein, or they don't react with that form of the protein in the cell. You're always going to get non-specific binding, especially when you overexpress a protein. Sometimes you will pick that up in if you overexpress a protein, sometimes you'll pick that up in like the GAP-DH, like in the GAP-DH blot, because you've just got so much protein in there. It's just, it's thinking that's a signal. But obviously, I think that just comes to like, if you know what size your protein is and have your like positive controls on the Western blot and a negative control as well, if it's relevant, that will really help you understand what the specific signals are.
Great, thank you. And I think one final one from my colleague Sritu, who's asked, do you see any differences in signal to noise ratio when using longer wavelength dyes in comparison to shorter wavelength? For example, 488 versus 700.
So I've actually only tested 680 and 800 in the lab, so I wouldn't be able to answer that question. I don't know if I'm trying to think what our LICOR can actually do, but all I know is it's 2-channel. So yeah, we've really just stuck with the 680 and 800, but we found them to be great for what we're trying to do.
That's really great. Thank you, James. If anyone else has any questions for James, please feel free to take down his email. It's been on the slides all this time. And also, you can see his Twitter handle here. Please do follow him on Twitter and also follow Protein Tech on Twitter, and just so we can engage in this conversation further and keep going. So that's it from James, and thank you so much for staying around. So I'm just going to give a little quick introduction to Protein Tech now. If I can go next. James, okay, brilliant. So just in case you're not aware who Protein Tech are as a company, at Protein Tech, we are the original manufacturer of antibodies, ELISA kits, and proteins. With our antibodies, we have over 13,000 human targets, and 2,500 of these targets have been validated by knockdown knockout validation. We acquired the nanobody company Chromatech in 2020, and they make leading nanobody reagents, including the market leader GFP trap for immunoprecipitation of GFP tagged proteins. We also have our humankind range, and humankind range are human cell expressed bioactive proteins, predominantly cytokines and growth factors. And also at Protein Tech, we have 5 global inventory sites, meaning that we have next day delivery for the whole of Europe and the USA. Can I do that? And so at Protein Tech, our slogan here is from our bench to your bench. And what we mean is that you can only ever buy Protein Tech products directly from us, and we never go through anybody else. And so from this, we have 100% self-manufactured antibodies, meaning that we can guarantee lot-to-lot consistency and also provide you with the original validation data. At Protein Tech, we celebrate our success by your success, so specifically helping you publish impactful research faster. And over our company history, you can see we've been featured in more and more publication citations each year, and we actually reached the landmark 100,000 publications earlier this year, which we're incredibly proud of and proud of from you, our customers, as well. So just a quick chat about some of the antibodies that we have for fluorescent Western blots. So we actually have our correlite conjugated antibodies, and these are directly conjugated, meaning that you have no need for a secondary antibody, enabling you to streamline your workflow and hopefully preventing some things where you can either get some off-specific binding or things that can go wrong. And our correlite conjugates are bright and stable, and they are equivalent to the Alexa fluor dyes. And we actually have some incredibly popular antibodies. So our GAP-DH is one of the top cited antibodies globally, and we now have this conjugated to our correlite 488, meaning that it's ready to use in as a control in your fluorescent Western blots. We also have our protein ladders, which are also able to be used in fluorescent Western blotting. So I also want to introduce to you the concept of nanosecondaries. So this is from our partner company Chromatech. And nanobodies are the binding domains of the heavy chain antibody from an alpaca. So if you see your traditional IgG here from something like a rabbit, what camelids have is they have the specific heavy chain antibody, and the binding domain, or the VHH, when removed, can then become a nanobody which binds with incredibly high specificity and affinity to a 3D target. And what we can do is we can then conjugate these fluorophores to a nanosecondary, to a nanobody, and then these become a fluorescent nanosecondary. And these nanosecondaries are available in isotype specificities to mouse IgGs and also to rabbit IgG. And they are conjugated to just general Alexa fluor dyes, which you can see in the red stars here. So one thing that we can do with our nanobodies is it allows for one-step incubation. And so what we mean by this is that you can have simultaneous incubation of your primary and the nanosecondary. And so what this means is that you, again, have a streamlined workflow, but also you can get some specific binding here with minimal or no cross-reactivity due to the nature of the nanobody binding. So thank you for listening this far. And so for anyone that has attended today, we are able to give you a free correlite antibody and protein ladder for everyone who's attended. So please feel free to email your requests to events@ptglab.com, and we'll also send everyone an email with how to request these as well. So that's it from us. Thank you so much for listening. Please feel free to get in touch with either James or myself, and hope you have a really great rest of your day. All right, thank you everyone. Bye.